Hello, everyone. Welcome to session 4 of ALCF's Intro to AI-Driven Science on Supercomputers. Today, we're going to hear from Corey Adams, who's an assistant computer scientist here at ALCF, and he's going to tell us about modern classification networks. And then we're also going to have a science talk from a computational biologist here at Argonne and talking about AI and within bioscience as well. So with that, I will hand things over to Corey and we'll get started from there. You're muted, Corey, if you're speaking.
Okay, let's try again. Can you hear me?
Yeah, you sound good.
Thank you for telling me. Okay, I was saying that today we're going to use a real data set for our materials, but there's a bit of a hiccup. We had the data set in a storage area for our workshop we ran last week and haven't transferred it over to space for this project yet. So you're welcome to try to run this notebooks, but it's going to fail on basically the second input step where you get the data set. So I'm sorry for that. We're going to get this updated as quickly as we can, and we'll update on Slack. So today, I'm going to talk to you about modern neural networks. And by that, I mean residual networks. We've seen in the past couple notebooks and sessions how to do the fundamentals of machine learning, what classifiers are, what convolutional layers are. And today I'm going to show you the results of a paper that came out in 2015. It has about 130,000 citations so far since then, and it has really kind of revolutionized the field of computer vision when it came out. And since then, many things have updated as well. Modern neural networks are even more modern, you might say. But we're just going to focus on kind of the contents of that particular development. So residual networks. The origin of that paper came from a data set called ImageNet. If you're not familiar, there was this computer vision challenge every year called ImageNet, where there was a big data set, and it drove a lot of development in computer vision. There were about 1.4 million images to classify, and there were over 1,000 classes. So it's quite a challenging problem. These were regular images, RGB pictures of about roughly 200-ish pixels on a side. So not huge pictures, but not as small as the CIFAR-10 or MNIST data sets. These were real images. And people competed every year to see who could develop a new technique to classify things even better. This paper that we're talking about today won that competition when it came out by a striking margin. So let's get into it. I'll go through the notebook here. It'll be hard to follow along, but the first thing we're going to talk about is just this data loader. And next week's session is devoted to what this data loader is doing and how to process and load data on supercomputers. Don't worry about this too much today. I will just kind of run all of these things and get down here where I've got some images and labels. And this should return in a second. 1,000 labels. This is our path for where our data set lives. 1.2 million files in the training set and another 50,000 in this validation set. The images here, the natural shape for this data loader are 256 by 256. So fairly big pixels. This turns out to be a convenient number for computer vision networks because there's often downsampling operations, and being divisible by 2 can be a very convenient size image. It's a batch size of 128 with 3 channels, and that's red, green, blue. So I'll just take the first image and show it to you. This one is random. Here it is. Looks like a band with some funky hats. And I'm going to take this image and apply some convolutions to it as kind of a refresher for what we talked about last week. So we learned about convolutions in the previous session. Because they're so foundational to computer vision models, I thought it'd be worth it to talk about them again. So this image up here, when we look at it like this, compressed to a small region, is pretty clearly an image to us. But to a computer, it's really just numbers. This has 3 layers per pixel, RGB, which your monitor can use to display colors. Each of those numbers, again, or each of those pixels, again, is just a number, typically between 0 and 256. I normalize it here to 255 to put them all in the range of 0 to 1. So it's just, what percent green, what percent red, what percent blue for this pixel. And that's just an artifact of this plotting library. A convolution takes some smaller matrix, in this case 3 by 3, and it just applies it to every 3 by 3 window it can in the image. So in this particular graphic here, it's showing you what it's looking like applied to the top left of this image. And this is a dot product, so you get 9 times 0, 4 times 2, 1 times 1, 4 times 1 to the whole 9 pixel grid. You sum everything up, and that becomes your output. And then to get the next output in your output image, you slide this convolution over, the input over 1, but you keep this, we call this the kernel, the same. So the convolution kernel doesn't change, and you slide the image input region over, and you get the next pixel out. That's a single convolution applied to a single image. Convolutional layers in neural networks are more complicated. So I mentioned that input there was 3 by 3. So imagine if in the second image you have, RGB. These are kind of red, green, blue, but out of order. And this one's not really red. But you can apply separate filters to it. This one is 4 by 4. And the convolution filter in your layer always has the same number of layers as your input image. So you'll take this green one, it will scan over the green image. You'll take the blue one, it scans over this blue image in the middle, and this kind of brownish filter in the back scans over this purplish pinkish filter input in the back. You do the same operation as up here, where you apply it to all of your pixels as you're moving to get the outputs. And then you sum up those outputs for a final pixel out here. So you get 3 by 3, where this pixel is the sum of this filter applied to the top 4 by 4 plus the second filter applied to 4 by 4, third filter 4 by 4. And that gives you one output filter. Most of the time in computer vision networks, you don't want just one output filter, you want a lot. And that means you learn a second set of convolutions. So there's another set of filters here, yellow, red, pink, purple, that gives you a different output. And then these just get stacked together, where they don't interfere with each other, but they become the input to the next layer. This is adding a nonlinearity and a bias, which of course is essential for computer vision networks. But that's kind of the idea. Your input gets a number of filters applied to it to make one output. You stack up a number of different convolutional filters to produce multiple outputs, and that's how you propagate downward in your network. Okay, so just to show you an example of that, here is a convolutional layer in TensorFlow. So it's using this keras subpackage of TensorFlow. So I create this object, sample convolutional layer, equals to tf.keras.layers.conv2v. And I give it 3 output filters here, and the kernel size is 1. That means that instead of 3 by 3 up here, it's 1 by 1, or just a single value for each kernel. So this particular filter is just scanning 1 pixel at a time, and it basically has a vector of 3 values that it multiplies by RGB and gets an output vector, and it learns 3 different vectors of those filters to make 3 output filters. Okay, so I'll create that, and then I can apply it to this image. Ignore the car here. This is the previous time I ran it. Here we go. Here are our musicians with their hats, and the picture, I claim, is just as crisp as it was previously in terms of, it has information spread out in the pixel, but the values for red, green, and blue are now totally different. And it's giving us this error up here, clipping input data to the valid range for ImShow 0 to 1. And that's because we didn't actually set any constraints on what the output value would be. So I scale up by 255, but the values in these layers are random. So they don't actually have any constraints on what the values output could be. So it doesn't have to be between 0 and 256. It can be 400. It could be negative. So it clips it here. Okay, awesome. I see a question on Slack. How do we go from 4 by 4 by 3 to 3 by 3? This is a good question. So let's just look at this green filter here. It's 4 by 4, and there's 3 of the filters total. So the 4 by 4 is the size of the filter. The 3 is how many filters you're using. And that 3 has to match however many layers are in your input. So this has RGB channels or layers. It could be more, could be less. RGB makes sense when you're looking at real images. Okay, so we take 4 by 4 and see how many times that fits into this input image, which is 6 by 6. So you can fit it in the top left, you can fit it once in the middle, and then you can fit it once in the top right before you've run out of valid spaces to place that filter. So that gives you 1, 2, 3 spots across the top. And you get 1, 2, 3 going down to end up with a 3 by 3 stripe, output space for that filter. Okay, I hope that answers your question. So convolution layers, I did this one with a 1 by 1. This picture up here is 4 by 4. This one's 3 by 3. That parameter for your filter can vary. So here I've got one that's 7 by 7. And if I create it and run it on that same image, now things are starting to get a little blurrier. Things have been shifted up, down, left, right. The colors, of course, are different, but this convolutional layer is now taking a single pixel, and the output image for that pixel is the same input area, but now it's got inputs from a much bigger region. So it sees the original pixel space plus all the pixels around it up to 7 by 7 away. There's another thing about convolutions that's useful to know. There's some terminology when you start using the machine learning frameworks. These are the things that you see most commonly. So strides, padding, you'll see mentions of output size or bottlenecks. Padding is an operation that lets you fit your filters into a bigger region. So I talked about how you go from 4 by 4 to 3 by 3 here, but it doesn't have to be that way. You could start with this particular image, just overlapping this 4 on the top left and then zeros for everything else, and scan it over that way. So you get an output that's much bigger than 3 by 3. And the zeros are artificial, but you've basically extended the image by padding it with zeros. And that's what's meant by padding. Some machine learning frameworks will show paddings like valid, where you've applied it only in the valid locations, or you'll see things like same, where it adds padding automatically to keep your image output the same size as your image input. And sometimes in other frameworks, there's not nice names for it. You actually have to do the math to see for a given kernel size and stride, what does the padding have to be to make it valid or same or anything else that you want. I mentioned stride. Stride is the steps that your kernel takes. So this one here was just, I was talking about how it went over 1 pixel at a time to get 3 by 3. But it doesn't have to be like that. It could start 4 by 4 and jump 2 pixels. So instead of the top left image of the filter starting on 4 and going 4, 9, 2, 5, the next time it goes, it could go 2, 5, 8, 3 and just skip that 9 column. And then your output instead of 3 by 3 would be 2 by 2. That's a commonly used operation that can be used to downsample images. So if you want to make a smaller image in your output than input, the stride can be useful. Bottleneck is also a term you see a lot. That's just a special convolutional layer. It has kernel size 1 and stride 1. So that's actually what I described up here with the first image I showed. Here it is. The output filters was 3, but the kernel size was 1 and the stride was 1 automatically. A bottleneck is basically just taking an input image, and for every pixel, it applies a multi-layer perceptron to that individual's pixels filters. So in the case of RGB, you've got for every pixel, the RGB, the B, and the G values going into an MLP to give you some outputs. And you can have any number of outputs. You often see bottlenecks used to reduce feature spaces. So maybe you'll have 256 input filters and you want to have a big convolutional layer, but your GPU can't handle that. So you do a bottleneck to bring it down to a smaller number, do your convolution, and then do the inverse of the bottleneck to bring it back up to the original number of filters. This is a common technique to save computational time and GPU resources without much loss of precision. Okay. I saw a question on Slack that's a great question. I should have mentioned that. I've talked a lot about the mechanics of what a convolution filter is doing, but what's the thing that's actually getting learned? It's the filter. And in the case of a bigger convolutional layer, it's the set of filters, as well as the bias value, this B over here. So your machine learning network will learn the values of these filters to get applied to images to produce outputs. And the inputs are set, the filters are trainable values, the bias is a trainable value, and then you compare it to a loss function and can do a back propagation. So anytime you see a convolutional layer in a neural network, it's the filters that you're learning. Yeah, the filters are like the weights in a linear model. Okay, so that's a lot about convolutions. I hope it was somewhat clear. Please go ahead and keep asking questions on Slack. In the meantime, I'm going to talk about one thing that came up in the past. There was something of a case for ResNet, though, not entirely. It was called the vanishing gradients problem. So as people in the first half of the 2010 decade or so were building deeper and deeper neural networks, they quickly discovered this issue where the gradients they were doing in their networks were getting smaller and smaller at the beginning of their network. And one way you can think about this is that as you're going through your network, you apply a convolutional layer or a linear model. You'll see kind of the same thing too. Generally, sigmoid was a really common activation function, so you apply something like sigmoid. That maps outputs between 0 and 1, so you get something that's less than 1 in magnitude. And as you keep applying those, your network's output can stay in the relatively the same range, but as you're doing the back propagation, you end up with this chain rule multiplying gradients that are all with a magnitude less than 1. And earlier and earlier layers in your network, which are further from the loss function, get a bigger effect of that multiplication. So your gradients get smaller and smaller and smaller further back in your network you propagate. This is called the vanishing gradients problem. One thing that came about to address this was called a residual network, where someone came up with the idea that instead of taking some inputs, applying a convolution and activation and returning the output, let's train it instead and say, what's the difference between the input and output? I want the network to learn how much I should change the input rather than totally changing the input. And if you Google ResNet, the first result typically is the paper that came up with this. And they have lots of good images, as well as you can find all of their derivation of their architecture and such. They run their network kind of like this. They take the inputs, apply a convolution to the inputs, apply an activation to the output of that convolution. They actually do two, so they learn a second convolution and a second activation, but they apply that activation to the output of the second convolution plus the inputs. So this is called the residual. It's the difference between the inputs and the outputs of the network. So they're not just learning the how to change your inputs, but they're learning some tweak to the inputs at a smaller level. This turns out to be really effective at dealing with the vanishing gradient problem, though there were other techniques I should share. So just as a demonstration of this, I put together two totally fake neural networks. So the first one was this just a series of these dense layers I defined up above. And this looks pretty generic Keras code, just defining this class called dense layer. Here is one called residual layer. I do 100 of these for the dense layers. I do 50 layers for the residual layers. And the reason for that is because there's 2 convolutions in the residual layer and only 1 in the dense layer. So this is the same number of convolutions as each other, even though it looks like it's half as many layers. Okay. So I'll create those networks. And then this is a totally fake gradient calculation, but I use this mechanism in TensorFlow called gradient tape. It's just with tf.gradient tape as tape. My output is the images, and then I just apply each layer to the output. And this is just bookkeeping to make this easier. Output becomes layer of output for all of the layers in the regular dense convolution fake network. So I'm just chaining them all together. And then I compute a fake loss. And it's just the mean of the output. That's not a real loss function. This is just saying whatever the magnitude of the output is, that's the loss on average. Okay, cool. So this is totally fake in terms of doing any sort of machine learning, but it's mathematically valid. So I can compute the parameters just going through with a list comprehension on this list of layers. I get all the params for each layer, and then I can compute the gradients from that loss. So tape.gradient of the loss with respect to the parameters. I do the same thing with the residual layers. I won't step through all of these steps again, but basically identical. And here I make a plot. So I'll compute the plot, and I'm just computing a ratio for every layer. So just do a loop over all the layers and their respective gradients. And I compute this ratio for each layer. I take the maximum value from the gradients and the maximum value from the same layer. And I take the ratio and I take the absolute value of that. And if you get a chance to run this yourself, you'll see that grad and layer here have 2 indices. Grad is actually a list here, where it's the weights and the bias. So just looking at the convolution filters here, the weights, the bias is there too, but I don't plot it. So I take the absolute value of the ratio of grads and the actual weights, and you can see here, even on a log scale, it's basically exponentially declining. And halfway through the network, it goes off the plot. And if you're doing anything in finite precision, there's only so far down you can go in exponents before your machine will round that to 0. So, okay, after half of the network, the gradients went to 0. Any update we do to the network is really only going to affect the last, quarter of the layers of the network. Some of these weights, even though they're not rounded to 0, some of these gradients, even though they're not rounded to 0, are so small that they just won't affect the way the network is behaving. Here's the same plot for the residual layer. You can see that it too has this sort of steady decline, but it makes it a lot further. This is a pretty artificial example with a fake loss function, but you can see that it declines only at the layer 10 instead of layer 50. Okay. I see some questions on Slack, so let me take a look at these. Last week's model used model.trainable variables. This week's uses model.trainable weights. I don't think there's a significant difference there in TensorFlow. We can check that out. It should be the same thing. And another question, why are we looking at the ratio instead of just the weights? That's because the weights can take a variety of magnitudes. They should have just been all randomly initialized from the same rough distributions. But if the weights were bigger in one layer than another, I wouldn't have wanted people to see an artificial behavior in this plot. Okay, so the difference here, I think, is pretty striking. The magnitude of the gradients makes it much further. Of course, I don't think anyone has ever trained 100 layers of dense convolutions with a mean loss to do anything realistic. So this is not a real problem. And there are ways to get around the vanishing gradient problem. Even so, the authors of the residual net paper found that their ResNet architecture was able to train better, even as you went deeper, when regular convolutional neural networks got worse when you went deeper, even with other techniques to get around this. Okay, so my intention today was to have some in-class exercises if we could get the notebooks working.
Corey, the data transfer just happened. So
Awesome.
I'll post in Slack, but I pushed a change to the repo. So if you do git pull again, you'll get the new path to where the data is.
Perfect. So let's try to do that. Everyone can now get on. I think we should take the second half of this session for everybody to try and get on Theta GPU and running these things. We'll try to answer questions in Slack, and you can raise your hand and maybe I can we can ask them online too. Let me describe to you the in-class exercise I want to do. So one of the techniques to get around the vanishing gradient problem was to use the batch normalization layer. So you can go to TF TensorFlow.org in the API and find batch norm. TF.keras.layers batch normalization. So this layer takes it basically learns the behavior of your network. So when it gets input, it's doing an average of what that input typically looks like, and it takes your input and scales everything to have basically a mean of 0 and a distribution of 1. And so it keeps all of your outputs from each layer roughly the same. The in-class exercise I want everyone to do is to take this layer and apply it 3 times. So before all of the sigmoids. So I'll take the output and apply batch normalization to it before it goes into the sigmoid for the dense layer and the residual layer, and remake these plots. Since we have the transfer working, Paige, is it possible to send everybody into the breakout rooms to try and debug this and get it working?
Yeah, I think we can try that. How long do you want them in the breakout rooms?
Let's Arvind goes it in 20 minutes or so, right?
Yep, we have a break in about 20 minutes.
So let's do like 12 to 15 minutes in the breakout rooms, and
Okay,
then we'll come back for a few minutes and share some results, and then we'll pause.
that sounds good. Okay.
Thank you.
All right, we should all be back. Corey.
Hey everyone. Okay. Talking with the breakout rooms, the overwhelming consensus, I think, was that Theta GPU Jupyter Hub is not simple. But this after the second session's class, we're going to have just a Python script. So if you can get onto Theta GPU, even if you can't get Jupyter working, you should be able to follow the second half and make some progress on the homework. So let me share my screen just to show you the solution to the in-class exercise. It was both meant to be not a trick question as well as kind of a trick question. So here's how it goes. To add a batch norm layer, you just create the layer here, tf.keras.batch normalization, and you store it with a variable name, self.norm. And you just insert it here between the convolution and the activation function. So output 1 is now equal to the norm of output 1. And for the residual layer, you do it twice. So create 2 batch norm layers, put it in between the convolution and the activation, between the convolution and the activation again. And then you run it And here's the part of the question that was kind of a trick question. You see no change. And the reason for that, which I alluded to very briefly, is the batch normalization has to learn the distribution of your data. So the first time, it doesn't know what the distribution of your data is. If you apply the gradients here to the parameters a couple times, these gradients should start to level out. And that's because as it goes further, as it learns the distributions of the inputs to each activation function through the network, it will be normalizing the whole way, both forward and backward passes, and your gradients should no longer vanish deeper in the network. All right, I'll stop there for my first half of materials. We've got a couple minutes left. I'm happy to take questions. If there's no questions, now is a good time to hop on Slack and ask for help getting onto the GPUs if you need it or anything like that. If you can't get the data set, go for that as well. So I'll wait for questions. If there are none, I'll pass it back to Paige.
I have a quick question, Corey.
Yeah.
So it's just about the padding. You were talking about you can put zeros around the image for padding, so you can get, I guess, those corner bits of the picture more. But would there be a case where you put different numbers besides zeros, like, I don't know, like ones or 255 or something?
Yeah, you could. I suppose I haven't seen a compelling use case for that, and it's not common practice. Mathematically, it's not invalid. It's also not super valid to pad it with zeros either, because then you're basically ignoring most of the kernels, and your output pixel might have a different mean distribution over your entire data set when you pad it with zeros. So I'm not sure. That said, padding is pretty common, particularly in segmentation networks. When you have an image and you have a network that applies to it and it gets smaller and smaller for many steps, when you segment it, meaning pixel by pixel classification, it'll get bigger and bigger and bigger, and you want to get back to the same image size. So that case, padding is actually pretty useful. In many computer vision networks that aren't segmentation, they're just classification, padding isn't really that often used.
Okay, thank you.
All right. If there are no other questions right now that anybody wants to speak up for, we can just take a little bit longer break today. So we'll come back in 13 minutes at 4 p.m. Central to hear from our science speaker. So with that, I'll pause the recording and I'll see you back in 13 minutes. Okay, welcome back all. So now we're going to hear from an Argonne scientist, Arvind Ramanathan, who works here. He does really interesting work combining sort of HPC and biology as well. So I'm just going to hand it over to you, Arvind, and then please go ahead.
All right, I hope you can hear me. I'm sorry, I'm actually sitting outside in a meeting hall, so it's like a little noisy, but I do apologize for it. Let me share my screen, which will be, and I'm sorry, I didn't have much time to prepare for this talk, but I'm going to kind of give you a sense of what we have been working on, so you get a sense of, where some of our science is really headed. So this is actually a project that we started building the last couple of months or so, what we call as foundation models for complex biological systems and how we design them. The idea really behind this is to think of biological systems as essentially Legos. So for example, you have proteins, you have genes, you have various components inside of them, and those are embedded within organisms. And we use AI to kind of like design various types of functions and kind of engineer new types of bacteria or new types of, this ones that have new phenotypes in them. So in that context, this is actually a project that was funded by the biological and engineering research, environmental research from DOE. And this is about building these microorganisms that I just talked about, but we are going to use these Lego blocks in a very contained manner. And the idea behind is to say you want to make sure that these expressions are constitutive, which means that you don't want them to just break down after they're done with their function. You want to be able to detect and respond to certain types of signals, and you also want to have probabilistic response, which means that depending on the type of output, you can actually see a different response from each of the organisms that might be in that consortium. So this is like looking at ways to engineer new consortiums, and that has like various applications, including biomedical applications, also for climate change, and also for a bunch of other interesting bioengineering side of the story. But at the heart of this story is this idea that we need an integrative platform that allows us to build rapid engineering tools for biological parts. And these biological parts, if you think about your cell as a car, you have various parts in them. So the car parts include transcription factors, which basically tell you which parts of the DNA that need to be like translated. You have like biosynthetic enzymes and various pathways that you need to create variants of for like improving the products. You also have genetic parts, which might be like pieces of DNA and so on that kind of regulate how these things are like coming out. And you can do these experiments in a high throughput fashion in an entire cell without the need for actually like ingesting them and doing a whole lot of like cellular expression. And that one is controlled by our AI platform that kind of like inputs all these things into this closed loop experimental system. So the question now becomes, how do you think of biological hierarchy and information, especially in terms of implicit and explicit representation, and with what we call as genome scale language models? And I'll talk a little bit about that. And how do we understand the hierarchy of biological information at both the individual gene and protein level? The last part is about how to understand the generation of these new examples. So I'll talk a little bit about the gene generation project. So when you come to biological information in terms of omics data, one of the things that we do know about is that single genes, there's a hierarchy of how the genes are organized. So from a single gene, which is about 1,000 base pairs, all the way to humans, which is like 3 gigabase pairs, you have a very neat way in which the genomes are organized. And within that context, a gene basically consists of, a promoter, a start codon, and a stop codon, and the open reading frame is anything in between. And you have a collection of genes also that have access as, ORFs, which is basically, you can think of it as a simple virus, or you have an entire genome, which is the collection of various ORFs that are put together in some order. And you need to learn that from the biological data. And people have done a lot of work on this. I'm not going to talk about this because it's very well known. People use what are called as protein language models to come up with language representation for the 20 amino acids that are chained together. You can actually look at the attention mechanism to capture the interactions between the different amino acids, and various ways in which people have talked about using natural language processing techniques for protein sequences, from profold recognition to cell localization to DNA binding, as well as structure prediction, which you would have heard, which is AlphaFold, which everybody has probably even tried a little bit. But the idea behind this is that you have various tools at your hands right now that allows us to do certain things at much larger scale than what was possible. Obviously, this has resulted in a lot of protein engineering achievements. And again, we don't have to look at all of them just to know that there are examples of this that are ripped. But there are some implicit representational issues, which is that we know that the sequences that we actually have are coming from the central dogma, which means that you have the DNA that is transcribed first into an mRNA or a messenger RNA. And from the messenger RNA, you actually get the protein, which is the translated version of it. So in that sense, the sequence-to-sequence models are actually kind of interesting in the way that they've been constructed. So if you enter garbage, you're probably going to get garbage out. But there's no notion of how the evolution happened at the single nucleotide level. And this is difficult to test in the latent space because it's very hard to interpret what the latent space means in terms of the this one. So that's where we started introducing what we call as the genome scale language models, which goes beyond what we call traditional K-mer models. And they actually take care of this issue of representing each of these proteins or systems as genes. That means that we are actually playing with the nucleotide information. So this lets us model the nucleotides very explicitly but let's just capture the information across multiple scales, both at the level of the gene, all the way to the collection of genomes, by using this type of representation. So we have shown that gene language models actually capture individual gene information. So this is a data set of about 36,000 malate dehydrogenase sequences, MDH sequences. This is a very well-known enzyme. And one of the things that we did was took the genomes and gene sequences and said, can we actually look at them at this local level of understanding what happens as a single gene? So interestingly enough, we can actually look at the clustering in the data. And if you walk from one side of the graph to the other, like how my cursor is moving, you can see that the GC content is actually increasing in each of these cases, right? A second thing that we also observed was that the sequence length, the embedding learns something about the sequence length, where you start at about 850 nucleotides here, all the way to about 1,050 nucleotides at the top. So if you move up, you're actually increasing the sequence length. If you're moving from left to right on this plot, you're increasing the GC content. But none of this data was actually used for the training. We used the raw sequences as part of our training. And what we get out of this are this very biologically informative aspects of what the data tells us, including GC content, sequence length variations, as well as molecular weight and other things. So these individual level models can use to interpolate and even sample from them. And that's what we did, which is that basically we took these genome scale models and then said, okay, can we actually look at ways in which we can sample them? And if you sample them, you can actually generate new examples of these genome sequences or gene sequences that lets us actually fill in the gaps of evolutionary things that we have not sampled. So that's kind of cool if you think about the power of these models, which is that you can use them for interpolating between different portions of the evolutionary tree without having to suffer through this process of saying what we may have missed in terms of sampling. And if you take these generated genes and translate them into proteins, it's kind of cool to see that the structures actually share similarity, not only the tertiary structure, that is how the protein is organized, but it also shares similarity in terms of the key residues that are maintaining the structure of the fold. So that's kind of interesting from the point of view of saying, okay, you gave the model a bunch of genes, the AI basically interpolated it and understood what it was saying about these co-evolutionary factors, and then it is able to generate new genes that look like what happens to be a valid malate dehydrogenase system. So at this point, what we are doing in the lab is taking these synthetic sequences of malate dehydrogenase and trying to work with them in such a way that we can validate their function by running assays on them and so on. The other interesting portion of this is that it also found other structures that had no similarity to this original structure, but that also might function as a malate dehydrogenase. So it's kind of interesting from the point of view of saying, okay, I am able to interpolate the sequences, but also understand there might be new functions associated with this fold, which can be used for protein engineering applications much down the lane. And now we also said, okay, why not extend this to an entire genome? In this case, we actually looked at the SARS-CoV-2 genome. And here, what we did was we took this genome because it's very ideal in terms of the size. It's about 30 kilobase pairs. And the GenSlims that we actually built have the ability to model these implicit evolutionary patterns that we can get from the SARS-CoV-2 genomes by looking at patient-level data, for example. So this can identify which portions of the genome are evolving relatively faster than others. And it can also tell you something about the different portions of the genome and tell how they are coupled evolutionarily. And so we test this by prospectively trying to model the evolutionary trajectory and trying to understand how these things look like, especially prior to how the people at WHO or CDC are able to identify these new modifications. So in order to overcome the length limitations of these gene-based models, we actually build them using what is called as a hierarchical representation. So it uses a GPT-2-like model. These are generalized pre-trained transformers, which is a very powerful representational idea. And then they build on top a stable diffusion model that basically learns the context of how the different genes have to be ordered so we can actually generate a whole SARS-CoV-2 genome. And one way to think about doing this would be to say, okay, I can build this model on top of what is called as a foundation model on all sequences that are known, about 110 million sequences. And then you fine tune this on the SARS-CoV-2 open reading frames. And then you periodically retrain on new variants and sequences that are across the different sequence windows. And you can use the performance that you learn from these different trained foundation models. And then you calculate what is called as a semantic similarity score. That's basically you embed the score into a low-dimensional space and understand how different sequences are related. And then you can use that value to guess what is called as the immune escape. And then you also use the sequence log likelihood score, which says at this particular position, what is the likely nucleotide that is to be present. And you can use that for fitness evaluation. And then you can tie that together with OpenFold-like programs to kind of find out how it structurally resembles to the proteins of interest, and then do a bunch of stuff to kind of say, can we measure the epitope alterations of interest where the protein is undergoing a lot of structural changes, as well as understand the interactions between the protein and the various host proteins that we are interested in looking at. So that basically lets us flag interesting variants that we try to look at. And we can use this diffusion model to generate new examples. And you use the same protocol for like finding out whether this is a variant of interest or not. Again, not much data here, but we can do this in an unsupervised way. We can cluster all the variants that are known, including alpha, beta, gamma, Omicron, which is shown in blue. And you can see that very clearly there is like some very well-known distributions that basically capture this data. And on top of it, we can also find the relative distances of how these sequences are oriented in the latent space, and that kind of gives us an idea of how they are distributed. And then if you generate new sequences that look like SARS-CoV-2, we can also find many of them that actually fit the same phylogenetic tree, which means that the generation process is actually kind of respecting the rules that it evaluated from the data. So, in some sense, it's kind of cool from two angles. One is that we actually can look at how quickly we can like understand a new variant and capture its behavior. And we can also understand how quickly it is to capture what variants of interest we must be going after, especially for vaccine design and so on. But these models are very expensive to train. So it took us nearly 62 exaflops to reach to this point of how we got to the these models to train really well. And they scale really well on our ALCF systems, which is including Polaris and Perlmutter. And we have also done some experiments using the National AI Testbed, which is called CS2 or Cerebras 2 systems. And without much details, again, we can do this on much larger scales than that is possible. In order to train this algorithm, it took us nearly 10.4 hours on 4 CS2 systems that were networked together. And this is not an easy task today in terms of running these types of models. So with that, I'm going to basically conclude what I was talking about, especially in terms of what we have been doing. We have these foundation models that can learn across different data sets, including applications for function annotation workflows, completing the metagenome sequences, as well as understanding these generative models that guide us in terms of doing experiments. And that's a whole other area in the lab that we work on, which is to have robots do the experiments, but AI actually control how these experiments are run at scale. And we have AI-driven simulations that are also important for advancing the integration of how experiments and theory can go hand in hand, and automated platforms that can allow us for better experimental design. So this is basically a summary of what all we have been working on in the lab. And if you have any questions, please feel free to post it to me. All right.
Thank you. So we probably have time for just a couple of questions. We have one in Slack. Alfred asks, can AI understand meaning? Like structures of proteins seem to fall into the domain of meaning.
Yes. So that's a great question. So we are trying to understand if the AI is really understanding structure or really understanding the meaning of what it means. Those things are going to come back to haunt us when we actually do the experiments, I hope. So when we do the experiments, we'll know whether it is really understanding the meaning. So far, in terms of the reliability of these models, what we have found is of the 100 sequences that we sort of sampled, about 96 of them have the correct fold, and about 4 of them don't actually fold correctly, which is good news in my opinion, because that means that, at random, if you take a shot at getting these sequences synthesized and run, it might take us a lot of time to run these experiments. But hopefully that, the AI models are themselves like understanding some things that are interesting, and we can use that information further to like guide our experimental design. So yeah.
Cool. And so a question for me, what's sort of the bottleneck in this process then? Is it more on the experimental side than actually creating these like simulated potential proteins?
No, actually the bottleneck starts out with the computing end of the story. So really speaking, if we had an in-house gene building engine, which is possible, which is really doable, much of this stuff should not be very hard to do inside of a lab, right? Expressing the protein, purifying it, and so on currently is time consuming because it's done by humans, and we are asking people to do it through the through their true interest in science of saying whether these models are actually working or not. But if you have an automated lab that can really increase the throughput quite a bit. The bottleneck, though, as I briefly mentioned, it takes several weeks on about 50% of Polaris to train these models. That's non-trivial in terms of like running these systems. And so we have to get to a point where we can get these training runs to go very quickly. But the idea that we can actually train them as a foundation model and have them specialize for other applications is kind of cool again, because once you train them, you just have to like fine tune them on the applications of interest that you have. So, yeah.
Wonderful, thank you. Well, I think we've run out of time, but thank you for coming and sharing the work that you're doing. It was really, really wonderful to hear about it.
Thank you so much. Appreciate it. See you.
Of course. Okay, with that, we're gonna pop back in and I will hand things back over to Corey.
Page. Okay, I'm going to share my browser again briefly. I just wanted everybody to see this paper, Deep Residual Learning for Image Recognition. This is the paper I was talking about in the first part of today's class. And the thing I want to draw everyone's attention to is figure 3. Let me zoom in on this. Okay, so figure 3, 34-layer residual network. This is one of their networks. It's not the very best one in this paper. In fact, the very best one is a couple hundred, is maybe 100 layers, 150 or so. But this is the network we're going to implement in today's second half of the class. I'll show you how we do it. And it trains, getting to error rates of approximately, I don't know, 20 to 30% at the very end. Okay. That was the wrong button. So if you go back to the GitHub repository, you will find a script here, trainresnet34.py. This is an implementation of the script to train that network I was just showing you. I'm going to walk through this script a bit and then show you how to run it on Theta GPU. So it's no longer a Jupyter Hub type notebook thing. So this is something that you actually have to get onto the compute nodes and run. You have to, let's see, post the link to the paper. I'm sorry, didn't I post the link? Yeah, the link of the paper is above. Okay. So, like I did in the notebooks, I defined this class residual layer. It should look very similar. I've got the batch normalization built in, norm 1, norm 2 this time. And again, it's this conv 2D batch norm, conv 2D batch norm, and then addition operation. And in this time, instead of a sigmoid, I have this ReLU activation, rectified linear unit. I know we've seen that in previous lessons, so this shouldn't be too much of a surprise. But this is the basic building block of this layer, this network. Here is a different tweak of the residual layer. And in the paper, this thing that I'm calling the residual downsample block, it's this dashed line thing. And what's happening here is on these dashed lines, it's going from 128 filters at some resolution to 256 filters at a resolution that's half as big. So 512 pixels would become 256, 4 pixels would become 2 in this case. And the way it's implemented here is with a stride. So I mentioned this in the first half. This is a strided convolution where the stride is 2 by 2 instead of 1 by 1. And then there's also an identity filter, which is a bottleneck, like I was talking about in the first case. Instead of a 3 by 3 filter, it's 1 by 1. And again, it's stride 2. So this isn't quite the identity, but I'm calling it the identity because it's very close compared to some of the other filters. And instead of adding the output to just the inputs directly, I call this identity operation on the inputs to get X. And now X here is not equal to the inputs. It's equal to the downsampled version of the inputs with some minimal weights applied. Okay. So here's the network. I'll go through it with some level of detail, and please go ahead and ask questions as you come up with them. So I created an object called ResNet34, and it has at the very first layer an initial convolution. In the diagram, that's this box right here at the top, 7 by 7 column, 64 divided by 2. Right here, this has, it's using the Keras sequential tool, which lets you string together a couple operations at a time. So it's got a conv2D with a 7 by 7 kernel and stride 2. So stride 2 is going to downsample with 64 output filters. So exactly like it's described here, 7 by 7 column, 64 output filters, divide the spatial size by 2. And then padding is kept the same, and there's no bias applied here. It has a batch norm and a rectified linear unit, and pretty much every convolution layer in this network is followed by a batch norm and a ReLU. This particular combination of convolution, batch norm, ReLU is so common that hardware vendors, when they give you code like NVIDIA does, will actually fuse these into one operation for performance reasons. And then there's a pooling layer, like described here in this network, pool divided by 2. So I've added a max pooling layer here with a stride 2. Okay, and then the paper goes through a series of steps where, and I've taken my code, and the colors here are where I've broken things up in my code too. So it's doing all these kind of light blue, and then these light green, and then these light red. The first branch is all of these 3 by 3 convolutions. And this identity layer, if you look at it, it's 2 convolutions and then some with the identity. That's a residual block. So this is 3 residual blocks here as I define them. So I called it residual series 1, and it's residual layer, residual layer, residual layer. And again, just as a reminder, that's this class up here where I've got the 2 columns and the batch norm and the ReLUs all built in. So it's calling this 3 times, but it's making 3 separate ones. So it's not just using the same weights 3 times in a row, it's using 3 separate versions of this. Okay, then there's this residual downsample layer. That's this step where it's got this dashed line. And then it's 3 more residual layers, but now there's 128 filters. So that's residual series 2 here, with 128 filters on each one. And just like before, there's another downsample layer where it gets to 256 filters. This time, if you count these up, there's 1, 2, 3, 4, 5 residual layers. So I have exactly that in my notebook. Residual series 3, 1, 2, 3, 4, 5 residual layers. Another downsample, as seen here, and then 2 more residual layers at 5, 12 filter output filters. You see them here, and then there's an average pooling. This is a minor deviation from the network described in the paper to this network implemented here. The image size used in the data loader here is a couple pixels bigger than the crop of the image they're using here. So we're using 224 by 224. We have slightly bigger images. I had to increase this pool size in this average pooling layer to accommodate that. But this will come down in the end to an output from this final pooling layer that is batch size by one size images with 512 features per, in general. So basically, it's a dense output. It's a batch size by 512, and then there's this 1 by 1 single pixel image at each layer. This flatten operation turns that into what a dense layer can process. And so there's this dense classifier applied here, give you 1,000 output variables. And it's 1,000 because there's 1,000 classes in the ImageNet data set. So when you get to this classifier, you end up with the logits. All right. The call function for this network just goes through those things in order and returns the logits. So this is exactly everything that was described up here, just sequentially. Okay, so that's the network architecture. How do we train it, and how do we actually compute some of these metrics that I show in the paper? So the first thing that I was talking about in the paper was this accuracy. They showed this error percent. Error rate is 1 minus accuracy. So this is a function to calculate the accuracy. The logits, as it comes out of the network, is just a value for every class. So I take the biggest one, whatever value is the highest is the selected class. So I take the argmax, and then I just do a comparison. I cast them both to float values, and then I say, is the selected class equal to the label class? And then in all the cases that it's equal, it's a 1, and in all the cases where it's not equal, it's a 0. And I take the mean of that, and that's my accuracy. The loss function is this function here. It's a bit of a verbose name for a function. Sparse softmax cross entropy with logits. So I'll unpack this a little bit. Cross entropy you've seen before. That's a pretty common loss function in classification of neural networks. With logits means that there's no activation applied at the end of the network. So a lot of classification networks could apply a softmax to the logits so that the sum of all the classes in a particular instance comes to 1. That would be the softmax cross entropy function. When you do it softmax cross entropy with logits, you don't have to apply the softmax. And in fact, if you do, your network won't converge. And then this sparse term just means that instead of having a vector of 1,000 classes for each image, so the labels would be batch size times 1,000, you could just say, what's the single one that would be filled? So it's just batch size by 1, and that's the sparse component. And again, I compute the loss and take the mean. And that's the total loss of the network. To train the network itself, I use this training step, which takes a couple inputs. It takes whatever the network is, an optimizer, the images, and the labels. I use this same technique I used in the Jupyter notebook, except now I'm taking a real, actually valid loss. I use a gradient tape. I compute the logits. For the logits, I compute the loss comparing to the labels. And then I can take the gradients, where they're just the gradients of the loss with respect to the trainable variables. The optimizer applies the gradients, and I calculate the accuracy as I go. And I just return the loss and the accuracy from this function for tracking. Okay, that's the bulk of the training tools. The next step is this train epoch function. This function is a little longer, but it's not super complicated. I just take in what's my current step in the epoch and what is the current epoch, as well as the data sets for the training and the validation data, and then the network optimizer and this checkpoint thing, which I'll come back to, as well as the batch size. The batch size is just used to calculate how many steps per epoch for training and validation. So for each batch of data, which I get from the data set here, this train ds.take. This argument is how many batches to take. It gives me a train images and a train labels. I don't worry too much about what's going on under the hood in this step. That's the subject of next week's lectures. I perform the training step with those images, and then that's basically it for the training. Everything else is auxiliary. So I've got a start and an end time, and I just use that to compute how many images per second are going by, and that's used to measure performance. And I print out each step, what's the current accuracy. So this will show up on the command line, which I'll show you in a second. After each epoch, I save the model, so checkpoint.save. And after each epoch, I compute the accuracy on the validation data set. So that just prints out down here. Okay, let's skip the prepared data loader function for next week. Here's the main function. I get the data set. I can set these hyperparameters for batch size and number of epochs. 256 is what they used in the original paper. And then it just it creates the network. So just calls the constructor, gives it some sample images so that I can print out what the network summary is. You can see all the layers. And then I create the optimizer. Here I used Atom, and that's a pretty modern optimizer. This checkpoint tool is just a way for TensorFlow to manage how to save and restore networks. And then I call train epoch while the number of epochs is less than whatever you set. Okay. I'm going to switch over to my terminal, but I'm happy to answer questions about the network while I do Zoom shuffling. Hopefully you can all see my terminal. I am in the AI Science Training Series GitHub in this 04 Modern Neural Networks folder. In here, I've got trainresnet.py script. And pay attention to this one here, trainresnet34 underscore theta GPU.sh. Let's look at that first. Make this bigger. I hope that is big enough to read. This is a Cobalt script that you can use to launch this job without having an interactive node. You'll have to change the project. This is a project I'm on. You'll have to change it to the AICF AITP project. And then it just does this. It sets up the conda environment. So module load conda activate. It goes to my folder right now. If you try to run this, it won't work because you can't get to my folder. You have to point this to your folder. And then you just run Python train resnet34.py. This line here is not strictly necessary for the mathematics and the machine learning, but it makes things run faster on the GPU. So that's why it's here. All right. I can now run this because I'm on an interactive node, train resnet34.py. If you're following along, which is great, I encourage you to do that. So I'm going to ask questions here. You'll see some things print out. So the first thing is you get this error initializing and opening fabrics delays. Just go ahead and ignore that. This is printing out the configuration of the data loader, which we'll get into again next week. Yours is going to look slightly different. Mine's the non-updated GitHub that Bethany pushed earlier today. And then it prints out the number of parameters in the network. It should be about 21 million. Here it is. Okay. And then it starts training. So finish step 1 of 5004, epoch 0. It's giving me the loss and accuracy as it goes. So accuracy here is not scaled to a percentage. So this is 1.6 accuracy on the first try, and generally hovering around 1% as it goes. And it's training through about 600 images per second on a single GPU. If you pull up the paper and you do the math here, this is the original batch size they used in training the paper. So the original training went for about 50,000 iterations, I believe, or maybe it was 500,000, sorry. And they ran it with a batch size of 256. if you do the math with this throughput of about 600 images per second on a Theta GPU A100 device, that's going to take you about 2 days of running, which you can't do because the time limit for a single GPU queue is 1 hour. So don't expect to be able to train this network fully to convergence with the script as it is today. In the next few lectures, we're going to see how to make this go faster in multiple ways. We'll be able to go faster on data loading next week, so the single GPU performance will improve. And then we'll see how to run this on multiple GPUs, so you can actually go through bigger batches and get shorter epochs and faster convergence. For today, you should be able to run this for a while. In a single hour's worth of runtime on the GPU queue, you can get a full epoch done. I've seen the accuracy get to about 30 to 40% if you get to maybe 2 epochs here. It's not quite as good as the ResNet version that won the ImageNet challenge in 2015, but it's certainly far and away better than random guessing on these images. And until the ImageNet challenge, this was actually a very hard problem to solve. So it's kind of cool to be running the code that kind of revolutionized computer vision. So that's the network that we're running today. I'll leave my screen up in case you haven't been able to run it and you can kind of see what it's looking like. It's going to do this for 5,000 steps, but the accuracy will slowly increase, the loss will slowly decrease. Since it saves between each epoch, you can submit a non-interactive job, let it run for a while, and it will restore when you submit again. So if you submit another job without changing anything in the repository, it'll pick up the weights from the network that it had done in the first epoch and start the second epoch instead of starting from scratch. So you can actually string these together and get reasonably good performance of the model. The homework this week is to basically run this and try to plot how the accuracy and loss are changing as you run. So you can see my loss and accuracy are fluctuating a bit. Right now it's maybe 5% accuracy on average, so it fluctuates a lot. And the loss is sitting around 5. Sometimes in the high 4s, sometimes a little higher than 5. For this week's homework, I want everybody to try to run this for at least an epoch. And you don't have to watch it the whole time. You can see that it starts up and then go get a coffee or something or work on something else more productive than staring at numbers going by. And then when it's done, you can capture this text that it's got and just parse it in Python and make a plot of loss as a function of step, accuracy as a function of step. And there's no problem if you want to modify the script to make this printout easier to parse or even save it to a NumPy array. I have no issue with any of that. You can save the accuracy and the step and the loss as it goes. If that makes your life easier, by all means do it. But that's the homework for this week. I hope this was useful. I'm going to stop here and answer questions and try to help people who want to get on the system in our last few minutes today, actually get on the system and get it running. But I'll be on Zoom to answer questions as well. Let me check what's on slide.
Please, I have a question.
Yes.
Yeah, so for the homework, are we expected to like upload another file for the accuracy?
No, I'm not sure I understood the question entirely, but the homework is just to run this for an epoch or so. It doesn't have to be exact, and plot the loss and accuracy as a function of iteration. So x-axis is the step, y-axis is the loss, and y-axis is the accuracy. You can do them on the same plot, you can do them on a different plot. I'm not picky.
Okay, thank you.
Mm-hmm. Don't expect to get the same accuracy they show in the paper after one epoch. I mentioned that would take 2 days of running on the queue. And it's not worth doing that. we'll see in the next few classes how you can speed this up to much faster times. Records for training ImageNet are now measured in minutes typically instead of days.
We have a question from Alfred.
On Slack?
He's raised his hand. So Alfred, if you want to unmute, go ahead.
Yeah, just go ahead.
Hi, Corey. I posted the question on Slack, but maybe you missed it. Just a simple question. I missed the first 15 minutes or so of your talk today, and maybe you said it already. So how is, what are you talking today different from the first two classification exercises? I think the first one was using MSIST. The second one was using TensorFlow to work on pixels and stuff like that. So how is today's work different from the other two works?
Yeah, if I could use an analogy from what my kids are probably playing with, the MNIST data sets and the early networks are kind of like Hot Wheels cars. You know, they're small, they're fun to see how a car works, and you can't actually do anything practical with them. ResNet and ImageNet are much bigger. This is a real thing. This model was, takes 2 days to train instead of a couple minutes on your laptop. The data set is hundreds of gigabytes instead of a couple megabytes. This is the real thing instead of just example models. And we'll see as we go on, if 2 days sounds like a long time to train to you, that it sounds like a long time for me too. Part of doing AI on supercomputers is figuring out how to take real models on big data and go really fast and train really quickly. So this is kind of the first step in that direction. I hope that answers your question.
Yes, thank you. So what's the rest for today? You mind me asking?
No, I didn't want to cover too much today. The goal was to cover ResNet and try to get everybody on a GPU and running it. So the rest for today is to try to get on the GPU yourself and get this working.
Okay. Well, I'm on vacation. I'm driving from Texas to Louisiana.
So maybe
I just
you're done driving.
Yeah, I just make a long pit stop in a gas station somewhere, trying to participate. So I better sign off and get on the road.
Great, safe drive.
Yeah, okay. I'll talk to you guys later.
Thanks.
Bye.
If there's more questions about the science, please go ahead.
Yeah, there was one question in Slack from Jia Roy, who just asked, could you remind me what the code for continuing the stopped training was, which you think you did earlier?
Yeah, I flashed something by, I think. So I hit Control Z, which interrupts my process. It's not actually stopping anything, it's just pausing things. And then I was looking for code. I used this tool called Micro. So I see you wrote macro on Slack. This is equivalent to saying bin train ResNet 34.py, just to look at the code. The actual part that stops the training, well, it can stop anytime, but if you get to this line here, checkpoint.saved, it will snapshot your model. And that happens after it finishes one epoch. So if you can get one epoch done, you can save this, and next time you start it, down here in the main function, it will try to restore the latest checkpoint. So this line here, tf.train.latest checkpoint, will look for, okay, do I have any saved snapshots? And if it has one, it'll restore it. And that will restore the stop training. What I was doing was just flashing by, and now I have the problem how to quick thing. And then FG is the command to put whatever process you interrupted back in the foreground. Okay, so I see in the code for the code is for step in epoch that assign 0 to make it step in epoch that assign 1 and save. When you restore the model, if I understand the question right, you asked about when you restore something, it will restore this step in epoch and epoch variable. And then the actual loop is assigning, I think it's actually assigned add 1 to increment the step in epoch. I hope that answers the question in Slack. Perfect.
Oh, when I try to run the notebook from earlier, and I run
Mhm.
this get dataset method, I get a error saying that the keys and values can't be empty tensors. Is there something
Yeah.
to change for that?
We've seen that error a couple times so far. And if you get it in the notebook, you're going to get it in the script too, I think.
Right. Yeah.
I didn't get a chance to finish debugging that, but I think we need to pick that one up on Slack and keep working at it. I know
Okay.
Ricardo is looking at it too. I'm not sure what's going on. Hopefully we can sort it out offline and get the script running.
Okay. Thanks.
Yeah, I don't have an update on that bug yet.
Okay. Thanks, Ricardo.
All right, I think we can probably just end things about 10 minutes early. Corey, I guess are you willing to stick around for about 10 minutes just in case something comes up? But I think if things are going well for you, then I think you can sign up for today and we'll sort of go from there. Does that sound good?
Actually, Paige, can I add one more thing before we go?
Yes,
Share my
please.
screen again.
Yeah.
I mentioned people can do batch job submissions. Let me just cancel this. I'll hit exit here, but I wanted to show how to do this. Okay, so this is the login node for Theta. This script here, train resnet 34 GPY, can get run by this bash script, train resnet 34 theta gpu.sh, and you do that with the q sub GPU command, train resnet 34 theta gpu.sh. I just wanted to show this so that everybody knew how to actually run this. This will run the script in a way where you don't have to babysit it. So if the queue is full, it'll just wait for its turn in line and then run. And if the queue is open, it'll go right away. You can check your job with qstat GPU that you, whatever your username is.
If I try it that way, I get an error that it's trying to assert that the training file list exists, which makes it sound like there's a path that I haven't set. Is there a workaround for that? Like, is there something I need to change to the script? I changed the line in the script that says you have to point this to your local copy to point to my local copy as far as I can tell, but I'm still getting that error.
Okay, maybe paste that error on Slack in the chat for this class so I can take a look at it. I'm sorry, I'm a visual person. I gotta read the error.
Will do.
Thanks. Okay, thanks, Paige. Now I think we can go, and I'll stick around online for a few more minutes to try to help people debug.
Excellent. Okay, thank you. All right, so hopefully we will see you all next week. Obviously, as questions continue to come up, please post them in Slack, and we'll see you then. Bye all.